Today, we're going to dig into a really fascinating puzzle in the world of cancer treatment. We're talking about something called diffuse large B-cell lymphoma, or DLBCL. Now, it's the most common aggressive lymphoma. And while our treatments are pretty good, there's this huge question. When the cancer comes back, why does the timing of that relapse completely change the game? So let's get into it.
All right, first things first, let's start with the good news. And it really is good news. For the majority of people diagnosed with DLBCL, the standard frontline treatment works. You're looking at more than 60% of patients achieving a long-term cure. That's a fantastic number, and it's what everyone is aiming for. But of course, that's not the whole story. For that other chunk, somewhere between 30 and 40% of patients, the disease is either refractory, which just means it didn't respond in the first place, or it relapses, meaning it comes back later on. And for this group, well, the journey gets a whole lot tougher. So this brings us right to the heart of the mystery. Doctors have noticed this for years. When the lymphoma comes back, some patients respond incredibly well to the next treatment, while for others, it's just much less effective. And the big variable, the thing that seems to predict the outcome, is when it comes back. But why? What is actually going on?
So, let's try to crack this riddle, because it really is a tale of two completely different outcomes, and it all comes down to the calendar. And this is where it gets really stark. Just look at this data from the study. It lays out the problems so clearly. If a patient's lymphoma comes back early, we're talking within 24 months of diagnosis, their odds of responding to the next line of chemo are only around 35%. Not great. But now look at that bar on the right. If the relapse happens after 24 months, the response rate more than doubles to about 75%. I mean, that is a huge, huge difference. So what's the biology behind this? To figure this out, researchers basically had to become genetic detectives. They decided the clues weren't just in the patient's chart, but hidden deep inside the tumor cells themselves. So they sequenced the DNA from the very first tumor and then again from the one that relapsed.
Okay, so this brings us to a really core concept you gotta understand. Tumor evolution. Yep, just like animals, cancer changes over time. Its DNA mutates. This can happen in a straight line, linear evolution, where the new tumor is a direct descendant of the old one. Or it can be more like a fork in the road, what we call branching evolution, where two tumors split off from a much older, common ancestor.
Let's make this super clear with an analogy. Think of linear evolution like this. The relapsed tumor is the child of the original one. It survived that first round of chemo, learned from it, and grew back tougher and more resistant. Branching evolution? That's totally different. In that case, the original and relapsed tumors are more like cousins. They share a distant grandparent cell way back when, but they grew up completely separately and developed their own personalities.
So which path does relapse lymphoma take? Well, the answer the researchers found solves our riddle of early versus late relapse. And it really shows, brilliantly, why timing is absolutely everything.
Okay, here's the first half of the big reveal. When that lymphoma comes back early, its DNA is practically a mirror image of the first tumor. It's the same bad guy. It's that tough, resistant population of cells that just weathered the chemo storm and grew right back.
But, and this is the kicker, when the lymphoma comes back late, its DNA looks totally different. It is loaded with new and unique mutations. It's not the same tumor making a comeback. No, it's a completely new tumor that evolved on its own from a distant, shared ancestor. It's the cousin, not the child. And check this out. The researchers found this beautiful, direct link. The more time that passes between that first diagnosis and the relapse, the more genetically different the two tumors are. It's just a clear, straight line. More time equals more genetic change. Simple as that.
The study's authors really nailed it in their summary. They said that these late relapses are a genetically distinct and chemotherapy-naive disease. Now that phrase, chemo-naive, that's the golden ticket. It means this new tumor, it has never seen chemotherapy before. And that explains exactly why it's so much more sensitive to it.
So this is all fascinating science, right? But what does it actually mean for patients sitting in a clinic or for the doctors trying to figure out the next best step? How does this change things on the ground?
Well, this table lays out the new playbook. For an early relapse, we now know we're fighting the same resistant tumor. So hitting it with the same kind of chemo probably isn't the best idea. This is the moment to think about newer therapies, like CAR-T. But for a late relapse, we're looking at a new tumor, one that's chemo-naive. And that means that standard chemo might actually work really well again, just like it did the first time.
And this all boils down to one absolutely critical, non-negotiable action item from this whole study. If a patient has a late relapse, you cannot just assume it's the same old disease. You have to do another biopsy. It is essential. That new piece of tissue is going to tell you the genetic playbook of this new tumor, and that lets you make the smartest, most effective treatment choice possible.
So what this all adds up to is pretty profound. It's really a new paradigm, a whole new way of thinking about lymphoma relapse that gives both doctors and patients so much more power. So let's just quickly run through the big takeaways. Number 1, relapse timing isn't just a number on a chart, it's a powerful biological signal. 2, an early relapse is the original resistant tumor coming back for round 2. 3, a late relapse is often a totally new cancer, one that's surprisingly vulnerable to old treatments. And finally, knowing all this lets us get into truly personalized medicine, picking the right weapon for the right enemy at the right time.
And that leaves us with one last kind of mind-bending question for the future. This research is an amazing example of mapping cancer's family tree after it has already grown. But what if we could get ahead of it? Could we one day look at that very first tumor and predict which way it's likely to evolve, letting us cut it off at the pass before a relapse even gets started? That right there is the next frontier.